Hello, hi, good evening. So this is Krishnaveni. I am taking you through a journey of story of life on earth, and that's biology. So welcome all of you to my session. So I'm grateful for all of you who have joined my session today. So for all those who are new, for Monday, Wednesday and Friday, I teach for class 12, and for the remaining 3 days, that is Tuesday, Thursday and Saturday, I teach for class 11. So today is Wednesday, so we are talking about a class 12 chapter. So we're talking about the strategies for enhanced food production, and today is lecture 3. So we have had good, interesting, healthy discussion about enhanced food production, how what can be done, why it has to be done. So it's a pretty interesting chapter and it's pretty real too. So you can actually relate to what is happening and how to enhance food production. So that is all about this chapter. So this chapter is like a story that you already know and you're just revising, you're going through the pages. Okay, yes, this can happen. Yes, this can happen. So it has nothing much to do with your brain or stress, etc. It is a page that you can just turn about and recollect what is happening. All right. Okay. So before we go ahead with the topic today, so we are going to talk about plant breeding techniques, how plant breeding can be more helpful, etc. So before we actually talk about what is a plant breeding technique, yes, good evening Priyanka. So thank you so much for joining my session today. So I have few important announcements to make, few things to let you know, few things that you have to set a reminder to, and then we'll get into the chapter. 𝗵𝗵𝗶𝗀𝗃𝗃. Oh my God. So is this Keshav? Is this Keshav again? Hello, hi. Hello Vishal, hello, good evening, and thank you so much for joining my session today. So without you guys, the session is completely boring. So please don't use smiley. So if you use smiley, the operator will actually send a warning. So please don't use smiley. So without smiley, we can talk. How are you? Yes, I'm good. Thank you. You're asking me. So Arvi, hi, hello, good evening, and thank you so much for joining my session today. Yes. So you guys are 2 minutes late. Today I was 2 minutes early. Is that so? Yes, thank you so much, Vishal. So I always trust you. Okay, so that is Keshav right then. So you change your name every time. So yesterday you came in a different name. So today you're coming. So yesterday your friend Alex was in a different name and today you're in a different name. So do you guys decide beforehand? Okay, so today I'll go in this name. Is that so? Yes. So alright, so before we go into the session and talk about more interesting things, so please do like, share and subscribe, so that you'll get a lot of more notification and I have a lot more interesting sessions to come up with. Okay, so please do share the link, so let your friends also join. Okay, so before we go to the lecture, so that is lecture 3, let's see what I have for you. So this is one important announcement from Career Point, that is crash course for JEE Main 2021. I know you're all NEET aspirants, it's okay, just relax. But if at all you know a friend who's preparing for JEE 2021, please let them know there's a crash course going on, so you know what is crash course, right? So things go at a much faster rate, so we have no time for small talks here. So directly they talk about the syllabus and the important question. So listen, so they have an offer till 10th of January, so please do let your friends know that there is this offer, but after 10th you have to pay $1,000 extra. So let your friends know so that they can enjoy the benefit. Alright, so will you do this for me? If at all you know a friend, okay? Fine. So today there is a mentee as announced yesterday. No, this is a weird name. No, this is a weird name. So no for what? You're not Keshav. So this is a weird name. 𝗵𝗵𝗶𝗀𝗃𝗃. So you should have written 𝗵𝗵𝗃𝗃. Yes, yes, please. Please, Ryan, you can welcome. So he gave me the option as Ryan to call. Alright, so as announced yesterday, so today we have a mentee at 6:30 and the topic is applications of biotechnology. So we are going to play on 15 questions, 20 seconds each, and the mentee code is. So guys, please make a note of your mentee code. It's 9355133. So if you make a note of your mentee code, you can join well before time and you can reduce the confusion that you're having at exactly 6:30. Yes, Harini, good evening and thank you so much for joining my session today. Aman Verma, hi. So hello Aman. Thank you so much for joining my session today. So Keshav, please invite Alex as well. So the mentee code, please don't note down the mentee code. It's 9355133. Okay? Yes? Okay, you came to know. All right. So you can have all type of guesses that I am from Tamil Nadu, I am from Kerala, I am from Karnataka. I don't mind. So you're inviting Keshav into the mentee, right? Because we're going to talk about food. So please do join for my mentee, and it's up to you to get me 50 participants today. So today, no compromise. So if you don't get me 50 participants, there won't be any mentee on Friday. Yes, so shall we have this deal? So all of you who are right now watching, so please go ahead and share the mentee link. So bring in all your friends, your parents, etc. so that we can have a small game. hardly we are going to play for only 15-20 minutes. It's okay, so anyone can join. What our objective is to increase the number of participants. The one who is completely focused will play well, so that is different, but we also need some cheerleaders, cheer people, right, to motivate you. So if we are 50 people playing and if you're on top 10, you feel motivated, right? So that is the objective, so bring in a lot. Bye. कोई नहीं है. Hi, sorry. So now am I audible? Yes? It's okay. So there has to be some twist and turn so that all those who are sleeping will wake up. Okay, so now am I audible? Yes? So tonight at 9 p.m. you have a premiere, so do watch, and it's all about stem cells. So if you ask me, ma'am, is stem cell relevant? Yes. So as doctors in the near future, stem cell is one of the topic which all of you will pay attention to. So you have a lot of disease that are cured by stem cell therapy, so you will need to know what a stem cell is. So it's a very beautiful topic out of the box, and it will make you feel so magical because stem cell on its own is very magical. Alright, so do watch. It is not a very big video. It's a short dinner time video, and you can bring in all of your family to watch it because it is something which everyone should know. So what is happening in the medical field right now? So especially you as doctors should know, but also you can bring in your parents, you can bring in your cousins, it does not matter. It does not deal more with biology, but with the general field of how your stem cell can be used in therapeutics. So please do watch this video and do like, share and subscribe, and let me know what kind of videos you are expecting in the near future. Alright, yes. So thank you once again for all those who are here joining me. So even Alex is here. So thank you so much, Alex and Keshav, for joining. Okay, so let's go forward and look into what's today's agenda. Yes, another very important announcement, guys. If you're a person who will tend to forget very silly things, so take your phone and make a reminder. So if you're watching my lecture in your phone, so please write it down or please keep a reminder in another phone that is present next to you. So tomorrow the class timing has been shifted. So for till the next announcement, so till further announcement, the timing of our YouTube class has been changed. So please do make a note of it. So every day since till now we have been meeting at 4 p.m. From tomorrow, we'll be meeting at 3 p.m. sharp. So the class has been preponed from 3 p.m. to 4 p.m. So don't come and tell me, ma'am, I forgot. I thought the class was at 4 p.m. No, the timing has been preponed. So our meeting will be from 3 p.m. to 4 p.m. So please set a reminder. Yes, you will get a notification at 2:30 as well as at 3 sharp. But still make sure you remember the class has been preponed from 4 p.m. to 3 p.m. So tomorrow we'll be dealing from 3 p.m. to 4 p.m. Don't come at 4 o'clock when I'm saying bye. Don't come and say hello, ma'am. Alright, so I'm informing you again. So the class tomorrow is at 3 p.m. So until further announcement, it will be at 3 p.m. Alright, yes. So 3 p.m. to 4 p.m. Does anyone fall asleep? From tomorrow you won't sleep. You'll either sleep from 2 to 3 or from 4 to 5. So 3 to 4 we will all study biology, and tomorrow it's an interesting thing. So tomorrow we are not talking about any new chapter in class 11. Instead we are going to solve questions. Remember, I told you I'll bring about a question session. So tomorrow we will solve questions. Alright, so tomorrow we'll meet at 3 p.m. So thank you so much, Vishal. Yes, great. For daily or only tomorrow, so till I announce further, so till next class when I announce further, the class timing has been shifted. So until further announcement, it is at 3 p.m. Okay, for a short period. So remember, it's at 3 p.m. Okay, yes, good evening and thank you so much for joining my session today. Okay, so Keshav is not well, so he's going to a hospital. Okay, so ask him to take care. Okay, fine. So please make a note of this announcement. Don't forget. Okay, so from this announcement, if you're not present at 3 p.m., I can understand. Okay, so you have a memory problem. Okay, so tomorrow let's see how many of you present right here have a memory issue to remember the timing. Fine. So how many of you are technically having presence of mind right now? So that will also be noted in tomorrow's session. So if you're technically present and if you have a presence of mind and if you have made a note, so tomorrow sharp at 3 p.m. you'll be present. Alright, yes. So question from which chapter? So for the first unit in your class 11, that is diversity in living world. So you have 4 chapters, right? Your living world, your biological classification, plant kingdom, animal kingdom. So we'll solve the important questions as well as I'll give you tricks how to remember the examples. Alright, yes. One shot revision series. So one shot revision series of what? I've neither completed the portions. So how do I go for one shot revision series? But by the way, Daniel, thank you so much for joining my session. Great. Okay, so we'll first do unit-wise question session, unit-wise tricks, unit-wise important questions. Side by side, we'll finish the syllabus. So once I'm done with almost 10 chapters, then I'll give you one short revision. Okay, so then we can go for revision. First, let me complete the portions. All right, yes, great. Okay, so shall we go forward? So all the important announcements I have made. Remember, there's a JEE crash course going on, so let your friends know about it. And we have a mentee at 6:30 p.m. and you have a very interesting lecture at 9 p.m. So do watch while having dinner. Okay. So mentee timing is not changed. So mentee for today is at 6:30 p.m. So if we are having a mentee on Friday or not depends on the participants that you're bringing in today. All right. So as for now, today's mentee is at 6:30 p.m. So do join, and I've given you the code as well. And watch my video at 9 p.m. and the next important announcement is tomorrow class is at 3 p.m. Alright, yes? So great. So shall we go into strategies? So today also we'll not be able to finish the chapter because we have to talk about plant breeding. So today is when I'm going to introduce plant breeding. So we'll go slowly because it takes time to understand, because again when you're going to talk about crossing, it again goes to the genetic level. So we'll go slowly. In the next lecture we'll probably end this chapter and we can have a question session for your class 12 also. Great, yes? So all well and good. So no confusion with the announcements. Yes. Shall I go forward? No doubt everyone is clear. Yes. Shall I go forward with your topic today? That is strategies for enhancement in food production. So today I didn't get a lot of fish pictures because yes, Kesha was also not well. So it coincides. So today we're going to talk about a lot about plant. Yes. So Vishal is pretty active. So thank you. So someone said from 20th January the school is going to reopen. So great. Yes. So are you all in the mindset of going back to school? Or are you all comfortable in the online mode? You're pretty comfortable, right? You're sitting in your own comfort zone, listening at your own pace. You can pause, you can rewind, you can fast forward, you can increase the speed. So I guess all of us are pretty acquainted and pretty comfortable with the online mode. Or do you want to go back to the offline mode? Yeah, maybe 1 or 2 days you would like to meet your friends, have lunch together, pass comments, etc. But I think all of us are pretty quite comfortable with the online mode. Do you agree with me? Yes. How many of you want to go back to the offline mode? Yes. Actually, online mode has made us pretty lazier. I agree. But still, it's all the more comfortable, right? Yes, ma'am. Safety also. Yes, exactly. So you're at home, if you're hungry, you can eat anytime. It does not matter because I don't have to look at your video. Yes. But online school class is not much good. Okay, so Priyanka says she's comfortable with online class. Fine, great. So as long as you study, as long as you benefit, so everything is fine. Yes, I am because I miss my friends. Okay, so it is both pro and con. So it has both advantage and disadvantage. All right, so every coin has 2 sides. So it's okay. So let's see what a 2021 actually holds for us. So Priyanka says online, she's pretty comfortable. So great, okay. So what are the strategies that we were talking about? So we have already started this chapter. So we spoke about animal husbandry. So Vishal says online again. So great. Okay. So all those who are online will remain with me till your NEET exam. So that's great. So animal husbandry, plant breeding. So today we'll talk about plant breeding. Still we are left to talk about your single cell protein and tissue culture. So today we'll try to talk about your plant breeding, but I don't think we can finish. So we are done with your animal husbandry. I guess there is no doubt in animal husbandry. Then today we'll talk about your plant breeding, and these two will finish off in the next lecture, that is on Friday. So by this week, this chapter will be done, and from Monday we'll start the question and the short trick session. Okay, fine? Okay. So going forward, animal husbandry. So what did we talk about in animal husbandry? So management of farm and farm animals, that is your dairy and poultry we spoke about. So how to maintain and handle your cows, how about the safety of the cows, how about this hygiene of the cows, etc. If you remember, we were talking about this. And what is poultry? It is a group of domesticated birds, right? So on whom we depend upon for meat and eggs. So they come under the poultry. Again, you have to be careful about the quality that you're selecting, the breed that you're selecting. They should be very resistant to disease, because right now, if you know, once again, I'm saying there's a virus that is spreading among the crows. So please be safe, because you don't know what kind of variant the virus is, and viruses tend to mutate faster. So we don't know what kind of mutation it is, what kind of symptoms it will cause, what will be the effect on human. Already we are amidst so much of chaos, and this is a new scene entry, and it's a new hero that is entering. So that is the problem with your poultry birds. So please avoid eating meat right now, because until and unless WHO gives us a red alert, or gives us a safety measures or a advisory effect, so please avoid eating meat. All right. So I will miss you even after meet. Yes, I will miss you all too. You were such an interactive batch, which I loved taking class to. All right, yes. So then we spoke about animal breeding. So we spoke about two types. What are the two types of animal breeding that we spoke about? We spoke about inbreeding. Then we spoke about outbreeding. So what was inbreeding? When you mate or when you breed two closely related individuals, so that is called as inbreeding. So you do that for quite a long period of time until you enter a homozygous condition, that is a pure line, right? So what are the advantages of your pure inbreeding? So you get desirable characters. For example, among the males you choose a superior male, and among the females you choose a superior female, and then you mate, and the resulting progeny is also superior, right? So what is superior male? So superior in case of male can be a bull that has very masculine and allied features, and in case of female, that is a cow that gives us more lactation per eel. So you can select and mate, that is called as inbreeding. So inbreeding will directly select the desirable genes. At the same time, it will also eliminate harmful genes, because when the superior male and the superior female have both dominant harmful genes, they both will come into picture during mating, but your natural selection will not give place to the harmful genes. So as a result, the harmful genes will be eliminated. So that is also one advantage with your inbreeding. But one disadvantage with your inbreeding is your inbreeding depression. So continuous inbreeding will also lead to a problem that is called as inbreeding depression. All right, yes. So now next thing is outbreeding. So under outbreeding, if you remember, we spoke about three things. What are they? First one is your outcross. Then it is your crossbreed. Then it is interspecific hybridization. So what is outcross? So when you take out two individuals, so when you mate two breeds, okay, which do not have a common ancestor for 4 to 6 generations, so that is called as an outcross. So inbreeding depression can be overcome by an outcross. All right. So what are crossbreeds? When you take two different breeds, okay, for example, when you're talking about a dog, so when you take a Labrador and when you take a German Shepherd and you mate them, so they still come under the species dogs, but they're two different breeds under your species, right? So when you cross them, that is called as a crossbreed. And what is interspecific hybrid? When you cross two different species like your donkey and your horse, you get a mule, right? So when you have a male donkey and a female horse, you get a mule. So that is interspecific hybridization. All right, yes. So this is about outbreeding. So what is inbreeding depression? So when I said inbreeding, so I am mating breeds or I am mating individuals that are very closely related. For example, consanguineous marriages. So in family you have consanguineous marriages, right? You marry within relatives. So when you marry within relatives, you do not have a recombinant at all. For a long time, you have a very pure line. You have a same type of character, same type of traits that is prevailing in the family, right? So there is nothing new. At one point, what will become? It will lead to reduced fertility. because you are mating among the same group, so there is no chance of variation, so there is no chance of evolution, right? So it lead to what is called as reduced fertility, and that is called as inbreeding depression. All right, Rayan? Okay. So Illuminati is also here. So thank you so much, Illuminati, for joining my session. So this is what we were talking about in your animal breeding. So then we went on to talk about beekeeping. So the most common species is your Apis indica. So apart from honey, it also gives us beeswax, and we also spoke about the honey venom. So that was the new information that we learned in the last class, that is able to cure aggressive breast cancer cells, right? So what is the name of the venom? The venom is called as Militin, if you remember. So if anyone has read more about it, so good, that shows your interest. So next one is fisheries. So fisheries refer to catching, selling, also preserving, that is called as fisheries. So we spoke about your freshwater fishes as well as marine fishes. Yes. So I showed you enough number of pictures to highlight the importance of the examples that is given in your NCERT. All right. Yes. So you have a lot of pictures here. So going forward. Yes. So once again revising, what is honey venom? Okay. So the honeybee can secrete what is called as a venom. So it is a particular secretory substance, just like you have hormones. So it also has honeybee venom. Okay. So that is what is harmful when it stings you. So that honeybee venom has a particular component called mellitus. So that mellitus, when added in a particular concentration, are effective against your breast cancer cells, which means they can kill your breast cancer cells. So normally when you're targeting a drug for cancer, it tends to kill your normal cells as well, right? Yes. So when you use your honeybee venom, it only kills your cancer cells, and it is not harming your normal cells. All right. Yes, Rian. So bye-bye. So you're going to park now. So thank you so much for attending my session. So please do put a like, share, and then go ahead. So have a nice time in the park, and bye. All right. So in between, when we're talking about animal breeding, that is your crossbreeds and inbreeds and outbreeds, we were also talking about controlled breeding experiments. So when I'm going to breed between two species, how am I going to do? So what control do I have over them? So that is when we spoke about a controlled breeding experiment. So first one was artificial insemination. So when you artificially hormonate or inject the cow with a hormonal injection, so that it produces sperms for us. So the semen, is a liquid that contains sperms, so that is collected from a male that is chosen as the parent. So the semen that is collected can be injected into the reproductive tract of the selected female by the breeder. You can artificially inject it, or you can even store it for future purposes. Okay. So it can be transported in a frozen form to where the female is housed. So these days, freezing of your gametes is also possible. For example, in CCMB in Hyderabad, if you have heard about this, so it is a CSIR lab. So CSIR stands for Central Scientific Institute of Research. So if you have heard this term, so Central Scientific Institute of Research, so it's a group of labs funded by the central government. So under this, you have a lot of labs. So in Hyderabad, you have what is called a CCMB, Center for Cell and Molecular Biology. So they have frozen. So there they deal with wildlife also. So you know a lot of animals are into extinction right now, right? So if you have the last animal on earth, so you try to collect the gametes of that animal and you freeze it, and then to increase the progeny, you grow them under artificially observed condition. For example, the first tiger, the gamete that was frozen, the tiger was developed into an animal in CCMB. All right. Okay, so your brother is working in Chandigarh, so you might have an idea about CSIR lab. So great, good. Okay. So the next experiment that we were talking about is your multiple ovulation embryo transfer technology as a junior research fellow. All right, so that is great. So that is multiple ovulation embryo transfer technology, that is MOET, right? So what did we talk about here? So where you inject the cows, so normally the cow produces a single ova, but you inject the cow to produce multiple ovas, okay? So you inject the cow to produce large number of eggs. So you allow the egg to fertilize with the semen. So we have already collected the semen in the previous experiment. So you try to fertilize the sperm and the egg. So once the fertilization has happened, you wait till it enters the 6 to 8 stage, sorry, 8 to, it is 6 to 32 cells, I guess. 8 to 32 stage cells you wait. So once it has grown from 8 to 32 stage cells, you take it and implant it in a different cow. Not in the cow that produced eggs, but in a different cow you'll implant it. So this cow is known as a surrogate mother. Alright, so you implant the eggs in this cow and this cow will give birth to the young ones. But the calves are identical to this cow. Okay, understood? Ma'am, if you are going to fertilize only one egg, then why are we ovulating it to produce more number of ovum? The chances of fertilization of an egg and sperm are very less. Okay, so here the egg and sperm has fertilized. Sometimes it will die immediately after fertilization. Okay, sometimes it will not cross the first step of division. So that is why you take in a lot of eggs so that by chance, okay, you do not have any death and you have one proper fertilization happening. So that is why you super ovulate the cow. The sperms are always produced in millions, so that is not a problem, but the ova is always produced as one. Okay, so that is why you wait till the 8 to 32 stage and then you insert it or implant it inside a surrogate mother. Alright, so this is what we were talking. So going into today's topic, that is plant breeding. So we are done with animals. So long the agenda of the chapter is to talk about enhanced food production. So how to enhance the food that we're having, because we spoke about the population right now. So the population is ever increasing, but the resources are decreasing. But how to make a smart move in this? So how to manage with the limited resources that we are having, at the same time feed all the mouth and the stomach that is present in the world. So one way for food, we are dependent on plants and animals. So a lot of people are non-vegetarians, they rely on animals for food, animal meat and egg. So we spoke about how we can increase the production or the strategies that we can implement to increase the animal food, right? So now talking about plant. So plant is where a lot of people depend on. So be it non-vegetarians, be it vegetarians, so all of us depend on plant. It is not that non-vegetarians do not take vegetables, they also take vegetables, right? So plant breeding is much important and you have to pay a lot of attention to it. Yes. Whether MOET is harmful for the cow, so too much of overstimulation. So for example, the mother cow in the previous slide, so if you give so much of hormonal injection, so it's going to have a hormonal imbalance. Yes, to a particular extent it is harmful. So 1 round, 2 round, if the cow is very disease resistant, it can survive. But if the cow is normally weak, so we don't select the weaker cows. But yes, because you're giving a hormonal stimulation, yes, sometimes it is dangerous. All right. So changing the traits of plants in order to produce desired characteristic. Yes, so that is what we're going to talk. So how are you going to select? That is important. So Pallavi Rao, good evening. Yes, thank you so much for joining my session today. So Aman Verma. Okay, so Aman, Pallavi is your friend. So thank you. It is termed as plant. Okay. Yes, I read. Not exactly inbreeding depression, but hormonal imbalance can create a lot of other changes. So it can lead to the changes in your brain, because hormonal stimulation, it can even harm the uterus of the cow. It can make the cow even infertile. So many things are there. See, as such, the body is functioning, that is fine. The moment you touch it indirectly to change the balance, to change the equilibrium, then it is at danger. All right. So the same with IVF. Why or not everyone is going for IVF? That is the reason, because you're artificially stimulating. You do not know. Sometimes the hormonal injections can become steroidal and can in turn become cancerous as well. So there are a lot of side effects. There are a lot of consequences that you have to consider before a particular treatment. All right. Yes. So in a way, yes, it's also going to reduce fertility. So you can call it as depression, but it's not between, you cannot say between along the same breed. Okay, so it can be between 2 breeds, it can be outcross as well. All right, okay, fine. So going forward with plant breeding. So this is one important technique where we are going to use your genetic engineering techniques, tissue culture techniques, etc. Because it is one thing that you can actually enhance the production of food supply. Animals are still fine. Not the entire population of the world are going to feed on animals. A majority of them are going to feed on plants as well. So we rely on plants for fruits or for a lot of things, right? For vegetables, for the fiber that they give, a lot of economical importance of plants also we have discussed. So based on that, how can we increase the yield? How can we increase the production of plant? At the same time, how can we meet the demand of the ever increasing population? So that is when we went to talk about your plant breeding. So traditional farming. So if you do it in the normal way, you can only get a yield up to a limited biomass. So biomass is the living amount, the amount that you're looking for. Okay. So that is the limited biomass. You can only feed the biomass is the food for humans and animals. So the traditional way only gives you a less amount, that is not enough to meet the ever growing population. All right. So better management practices and increase in acreage increases yield only to a further extent. So even if you have better management practices, better fertilizers, better pesticides, and even if you have proper irrigation techniques, you are well aware of what kind of disease attacks your plant, everything is perfect, even then your yield increases up to a certain limit. It cannot be done in abundance, it cannot be done in large scale. So what do you do in such a condition? So that's when plant breeding came into picture. So plant breeding increased the yield to a much larger extent. Alright. So just like you had animal breeding, just like you had crossbreeds, so just like you had interspecific hybridization, your plant breeding also increased your yield to a greater extent. But what is plant breeding? So what is that we're trying to talk about? So have you heard about green revolution? So we have spoke about green revolution a lot when we're talking about the applications of biotechnology. So it led to the tripling of food supplies, right? So from green revolution, we understood green revolution alone is not sufficient. We have to go ahead with what is called as transgenic plants. So ultimately, I'm going to talk about your transgenic plants when you're going to modify a particular plant, when you're going to take two plants which have desired characteristics and we are going to mate them. We are going to pollinate them here. So that is called as your plant breeding. All right. So for plants, it's not going to preen, okay? So we are not going to induce a hormonal injection. You're just going to select one plant which is capable of giving you good yield and one plant which is very resistant to disease, and we are going to pollinate them. So plants, it will not preen. We are not going to give a hormonal injection. Okay, fine. So depending on the plant breeding techniques, the development of high yielding and disease resistant varieties in wheat, rice and maize were successful. So what do you do? With the help of your genetic engineering technique and tissue culture technique, so your plant breeding was completely successful and it also led to the development of high yielding and disease resistant varieties in your wheat, rice and maize. Alright. So this is how you actually work in lab. You grow the plants in a test tube and then you study them. Alright. Yes, exactly. So we'll talk about tissue culture also in the next lecture. So tissue culture is one important technique. You can say it is a tool for your plant breeding. So this is how we work in lab. So we put your plants in a test tube and you study whether there is a proper crossover or not. So we'll talk about all this. All right. Fine. So what is meant by plant breeding? So here is an exact definition of plant breeding. So it is a purposeful manipulation of plant species. Not just for time pass you're studying the plants, hence it is not easy. So it is a very tedious procedure. So you're purposefully trying to manipulate something for a purpose. Okay, so you have a feasible purpose and it should also be possible as well. So to create a desired plant type. So to suit your cultivation, at the same time, it should also give you better yield and it should be disease resistant. So to make sure all these criteria are met, you do what is called as plant breeding. Okay, to produce a plant that is best suited for cultivation, that is easy to cultivate. At the same time, they can give you higher yields, better yields than the original ones, and also disease resistant. So everywhere if you see animal breeding or plant breeding, so disease resistant is a very, very important thing. So today survival of fittest is what everyone is talking about. So if your plant, if your animal can survive the disease resistant variety, so it can be the disease resistant one and that is going to survive. So it is literally the survival of the fittest. Alright, so classical plant breeding, so a normal plant breeding is crossing or hybridization of a pure line. So artificial, then followed by artificial selection to produce plants with desirable traits that can give you good yield, nutrition and disease resistant. So plant breeding, so just like animal breeding, you are going to cross two different breeds. So in plants, you're going to cross two different plants, one with a higher quality of gene A and other with a higher quality of gene B, and we are going to pollinate them to give you a high yielding plant, a plant with high nutrition and disease resistant variety. Alright. So this was also found by Mendel. Plant breeding? No, not exactly. So Mendel only spoke about your genes. So Mendel gave us an insight as to there's something that is being passed on from one generation to other generation. So initially he named them as factors and later that the term gene was coined. So this was actually, plant breeding was actually started. So once your genetic engineering started, then people started working with plant breeding technique. So your father of green revolution, that is Norman E. Burke, he first started off with wheat and rice variety. Alright, yes. So this is what gave birth to plant breeding. Okay, so when I talk about plant breeding, say for example, I have a plant A and I have a plant B. Okay. So first thing, if I am going to pollinate them, so this is a superior variety, so say this is of a particular variety. Okay, say this is of variety A and this is of variety B, and in this you're choosing a superior female and a superior A plant and a superior B plant. Okay, now if I have to pollinate them, so is it enough if I just consider 2 plants? For example, if I want to combine a disease resistant plant with a high yielding plant, say for example A gives you high yield. Okay, so here we are taking your rice. Okay, for example, it is a hypothetical example. So we are taking rice. So in rice you have so many varieties, right? So at home also you have so many varieties of rice. The rice that you use for the normal white rice, the rice that you use for biryani, the rice that you use for making idli dosa, that is also different, right? So you have so many varieties. So for example, if I pick the variety A, so this has high yield. So this gives me a high yield. And I look into the variety of B, so this is disease resistant. So now I want to combine because I'll get 2 desirable traits, right? So disease resistant. So now if I breed them, if I pollinate them, so will I get a super character here? Yes? Yes, basmati is also rice. इससे 𝗵𝗵 वाला concept भी clear हो जाएगा. Yes, true. So basmati is also one of your varieties. So under basmati you had 37 varieties. So you had 37 types of basmati rice. So basmati is one type. So one type of rice. So under basmati you have 37 types. Okay. So what happened? It was native to India, but suddenly a US company patented basmati. So if you want to grow a particular type of basmati, you have to take the permission of the US company. So that is against the ego, right? But still, that is what is happening. So you have to be very smart enough, even though if it's your discovery, immediately if you do not go apply for a patent, you lose your right. Okay? Yes? Yes, you can modify as much as you can. So that is possible only with the birth of a genetic engineering and your tissue culture technique. All right? So what are the factors should be considered? So before we actually breed or pollinate 2 different varieties, though they are having good traits, you should consider certain points. Whether they are giving you increased yield, that is fine. Whether they are giving you improved quality, okay. So you should also consider this point that is very, very important. So let me erase this. So increased tolerance, so this also is one point that you have to consider. Increased tolerance to environmental stress. Is your plant able to tolerate the environmental stress, that is salinity? So depending on the soil, so depending on the salt content, the extreme temperatures to which place you're going to plant, and drought conditions. Alright. For example, if you're going to bring that variety of rice to Rajasthan, it should be able to tolerate drought condition, right? Yes. So all that should also be considered. Resistance to pathogens. So disease resistance is fine, but resistance to which type of pathogen? It should be resistance to your virus, fungi and bacteria, because these three, they shake the ground of your plants. Okay, so they cause enormous diseases, they affect your plants, they invade your plants. So that's why you should check. For example, if rice is affected, if your sugarcane is affected by bacteria, so that particular variety should be able to be resistant to bacteria. All right, yes. So Keshav, hi ma'am, I'm not able to talk, so I try to watch silently. Okay, so you are in your name. So who is the other one? So did you change your name and come in now? So all right, but still thank you so much. I heard you're not well, so please take care. So increased tolerance to insect pests. So when we were talking about your BT cotton, when we were talking about your tobacco, when we were talking about your RNA interference, so what was the main thing? What was the main drawback? That was the insect was eating your plant and thus decreasing the yield. Your plant as such is fine. But one problem is the insect come and eat the plants and your yield decreases. So that was the case of BT cotton. So that is why we went about to use your Bacillus thuringiensis gene, right? So your plant should also be resistant to insect pests, right? So Anu, good evening. So I guess you're new to my session. So thank you so much for joining my session. So please do like, share and subscribe. So I teach the entire biology. So today we are talking about strategies for enhanced food production. All right, yes. Can we make a superior human being? Yes, you can make, but you do not have ethical permission. So once you make a superior human, then you can do anything, right? You can clone. You can make another clone out of me. So how do you know which is the clone and which is the original me? So that will lead to ethical issues, but it's actually not possible. You have to take in permissions, but it's not allowed. It's legally banned. So if you make a superhuman, you will be arrested immediately and your superhuman will be damaged. Okay, so recently in Korea also the same thing happened. The scientist created a superhuman baby. He literally took the eggs of a female who was pregnant with a baby, and then he cloned it with another sperm that had a superior quality. So he literally made a baby in vitro. So that was not possible. So that is against your ethical model. Okay, all right, fine. Wah. So what are you trying to me? What are you trying to play with me? So there are two characters here. Okay, fine. Yes, cloning is possible. That's why I said, no, we are equal to God. We can create life. We can create human beings. But with greater power comes greater responsibility. So it becomes an ethical issue that is very, very legal. Okay. Thank you so much, Anu. Thank you so much. So I'm very grateful to your chain of friends. So please do ask them to come and watch my lecture as well. Okay, so these are the points that you consider when you're that you should keep in mind when you're trying to breed two plants of a desired quality. All right, only original Krishna knows me. All right, so no one is going to clone me. So I am the only one original piece that is present. All right, so I'm not going to be cloned. So don't worry. So cloning is not allowed, and anyway, it's not easy. So Dolly, the sheep that was cloned, so it was not in the first attempt. They took several attempts to clone. It is not easy to clone as well. So things are easier said than done when I'm talking about a genetic or a molecular level. Yes, you can clone, but it is not possible. So even in plant breeding, it is not easy. So here we see how and why it is not easy. So plant breeding, the main steps for breeding a genetic variety of a crop. So you have a particular steps that you should consider when you're going to when you're going to pollinate two different varieties. All right, yes. Okay, fine. So first, collection of a variability. For example, if I am going to talk about rice variety, so I have a lot of rice, right? If I'm going to talk about rice plant, I have a lot of varieties. So many varieties in rice. So first, I have to understand how many genetic variabilities are present. So first, I have to study the genetic variability. So when I'm talking about step 1, that is collection of variability, the first thing I have to do is study the genetic variability. So that is the first prerequisite that I should do. So I should also know about what are the pre-existing genetic variabilities. So pre-existing genetic variabilities with respect to your already present ones, right? So the existing genetic variability I should study that is present, and also I should collect and preserve the new varieties, the wild type varieties, the cultivated varieties. So all the varieties first I should collect. Okay, then I should analyze which is more superior, which is less superior, what is the pro, what is the con in this. I should analyze everything. So here you have a term what is called as germ-plasm collection. So what is germ-plasm collection? The entire collection of all the diversity of your alleles of a gene. Okay, so we are talking about the genetic variability. For example, when I say rice, so rice has a lot of varieties. So they're genetically variable. So a diverse collection of all alleles of a gene. So of the alleles of all genes, so you have a lot of genes. So when you collect all the information about the alleles of a gene of a given crop, so that is called as a germ-plasm collection. So what are alleles? The alleles are the alternating or the contrasting options present in a gene, right? For example, if you remember Mendel, so it is capital T and small t. So capital T is dominant and small t is recessive, right? So when you have all the information of alleles of all the genes of a particular crop, so that is called as a germ-plasm collection. So first you will do the germ-plasm collection. So you'll understand what are the varieties present, which variety is more superior. So what is the advantage, what is the disadvantage? So that is how you will study. All right. So that is the first step, that is the collection of variability. Fine. How can I get the timetable of Career Point YouTube lectures? For that, you have to subscribe the YouTube channel. So when you subscribe the YouTube channel, you'll get a notification. All right. So you'll also get the thumbnail in the previous night. For example, today if my class is at 4 p.m. So you get thumbnail notification last night. You would have got a thumbnail notification last night. So that is how it goes. And also if you're regular to my class, so from tomorrow my class is at 3 p.m. So 3 to 4 p.m. we can meet. And also if you are a NEET aspirant, so I guess you are. So please do join for my mentee quiz at 6:30. So if you subscribe this channel, you'll get my notification. All right. Yes, that was in pea plant. I'm just giving you an example of what are alleles. All right. Yes, so that is the first step in your plant breeding. The second step is evaluation and selection of parents. So now you have your germ-plasm collection. Now from your germplasm, you will choose which type of parents you want. So what are the 2 desirable traits you're looking for? So based on that, you will select the parents. You will select one parent and you will select another parent. Alright, yes. So the selection of parents should be such that it should be very compatible with each other. So that is the selection and evaluation and selection of parents. You will evaluate your germplasm collection. Okay, so which variety you want and what type should it have. So you will select your parent. Alright, so that is the step 2. Okay. I don't know. So I don't know what is that. I can't even read. Okay. Okay, 0 educational year. All right. Okay, so your 2020 was your 0 educational year. I don't know. So cross hybridization among selected parents. So first you have collected your germplasm. Now you have evaluated your germplasm and you have selected your parents. Now the third step is to cross hybridize. For example, take a parent A. Sorry, a plant A. So plant A has high protein content and plant B is very resistant to diseases. All right, so now you're going to crossbreed plant A and plant B. Fine. So how are you going to do? It's a very tedious process. First you have to take the pollen from plant A and put it on the style or stigma. You have to put it on the style of your plant B. Okay, so that is how the pollination happens, but it is a tedious process, right? But that is how you do pollination, and you wait for the offsprings to develop. So that is cross hybridization among selected parents. You have selected the parents and you're going to pollinate them. Now you have a lot of progeny, right? So you have a lot of progeny, but the success rate is very less. You have chosen parent A and parent B based on 2 traits. For example, if I take plant A and plant B, so this has increased protein and this is very resistant to diseases. Okay, now if I am pollinating them, so I get a lot of offsprings. Offspring A, B, see, I'll get hundreds and thousands of offsprings. Okay, but which one will have the characters that I am looking for? There might be so many characters in plant A, so many characters in plant B. So when I crossbreed, there's a lot of permutation and combination that can take place. But which one is the offspring that I am looking for? The offspring with this quality. Okay, so that is a major problem. So you will have hundreds of progeny coming here and only one of them will show the success that we are looking for. Alright, so that is the problem with your plant breeding technique as well. It is a tedious process, but still selection of offsprings is very important. Okay, fine. So selection and testing of a superior recombinant. So I told you have n number of offsprings and only one particular offspring will have your desired characters combined. So you will first select and test that superior recombinants which you are looking for. Again, that is a tedious process. So you will so once you identify the recombinant, so once you identify the progeny that has the characters that you are looking for, you will self-pollinate it so that you will have a lot in number. You will cross multiply, you will self-pollinate till you get a pure line breed. Okay, because that is what you are looking for. So once you identify the recombinant that you want with the desired characteristic, you will multiply the plant by self-pollination and you will do a pure line breeding as well so that it reaches a state of homozygosity. Alright, and the last step is you will test, release and commercialize the new variety. So now you have a recombinant. Now you will check if your recombinant is satisfying the criteria of all other points. So in the previous slide, I showed you a lot of criteria, right? It should have improved quality, it should be resistant to environmental stresses, it should be resistant to pests, etc. So now you will take your recombinant and check if it is actually working in the field. Okay, so practically if it's working. So this was all based on your theoretical knowledge. Now practically you will check. Okay, then you will release it. So in a small scale, in a small piece of land, you will cultivate them and see if it's practically working. So testing, you can do it in a small test tube as well. If you remember the first picture that I showed you under plant breeding, so you can use that to test as well. Then you will release it on a small farm scale, and then only after final testing only you will commercialize your new sapling. Alright, so it takes a lot of procedure. It is not easy. It will take months and years together. Alright, fine, great. So is this understood? Plant breeding is understood. So examples are your wheat and rice. So they tried plant breeding. So with the help of your rice and wheat, sugarcane, millets, so they all produced your hybrids. So we are going to talk about each of them in detail. So wheat and rice, they had semi-dwarf varieties. So let's quickly look into the examples and then we'll stop the class. So we still have to talk about your disease resistant and your pest resistant. That will continue in the next class, followed by your single cell protein and your tissue culture. So before that, let's look into the examples of plant breeding. So the first example is your wheat and rice. So in 1960 to 2000, so wheat production increased from 11 million tons to 75 million tons. So that was a huge increase, right, with the help of your green revolution. So then rice production from 35 million tons to 89.5 million tons, and still we are not able to meet the demands of the ever increasing population. But though it was a statistically good increase, right? Yes, it was definitely a good increase and it was possible with the help of your green revolution. So what led to this increase? They also used plant breeding techniques here. So they developed semi-dwarf varieties. So you had a tall plant, you had a small plant, you had a dwarf plant. You had a tall one, you had a dwarf one, and in between they produced a hybrid that was called a semi-dwarf variety. So Sonalika and Kalyan Sona, they are high yielding and disease resistant wheat. So they are semi-dwarf varieties. So who led to this semi-dwarf varieties was your Norman E., the person who was the father of your green revolution abroad. All right. So he plant bred these two types of varieties. So semi-dwarf varieties in India were Jaya and Ratna. So they were the two varieties. They are rice varieties. All right. So you also had high yielding varieties of rice. So this came from the international rice research from your Philippines, and this came from Taiwan. So they interbred both. Yes. So this is how you got the increased production of your rice and wheat. All right. Great. So next it was sugarcane and millet. So what has sugarcane to do with it? So in South, the sugarcane is pretty famous. Yes, your Pongal is coming, so you might know. So Sacrum Barberry is a sugarcane variety that grows in your North India. So it has a poor sugar content and it has a less yield. So it was completely a not desirable plant. But at the same time, you had another one in the topic that is called as the topical canes. And in South India, the variety is called a Sacrum officinarum. So this had a very high content of sugar, but it was not able to grow in North India. So as a result, the sugarcane has to be transported. That is still a tedious process, a time-consuming and a cost-effective process, a cost-consuming process, right? So Norman E. Borlaug is the name of the person who's called as the father of green revolution. All right, okay. So now they wanted to grow this variety, Sacrum officinarum, in North India, so that they also have a good variety. Okay. Yes, yummy sugarcane. Yes, even I like sugarcane. So your Sacrum officinarum has a thicker stem and they have a higher sugar content. So that is why we prefer your Sacrum officinarum. So what they did, they initially took out the genes from this and they tried to clone it so that they are able to adapt in North India. So basically your plant breeding techniques was performed. So after that, your Sacrum officinarum was able to grow in your North Indian conditions. All right, yes. Great. So similarly, your millets were also modified. So millets, so you had the production of your hybrid maize, jowar and bajra. So they were also developed in India with the help of your plant breeding techniques. So what was the highlight here? So here the highlight was it had a thicker stem, it had a high sugar content and it was not able to grow in North India. So they used plant hybrid techniques to grow it in North India. So what about in case of your millet? So they were water resistant varieties. Water stress resistant varieties were developed. Alright, so these are your millets, your jowar, bajra, maize, etc. They are called as your millets. Yes, so I don't know if the picture is clear. So check out how many types of millets are there and how many you have eaten. Alright. Yes, you're right, Aman. So which means you're clear with this. So great. So let me show you the sugarcane pictures. So yes, so Sacrum Barberry, so if you look, this plant itself has a very thin stem and it's not going to give us a good quality of sugar. The sugar is not as yummy as that, okay. But the one that grows in South India, that is your Sacrum officinarum, the normal sugarcane that you get during your Pongal, so that has a very high sugar content and it has a thicker stem also. So they wanted to grow this variety in North India so that the problem of transportation is reduced. So what they did, they plant bred. So they literally took out the desirable characteristics that is enabling this plant to survive in North India and they injected into this, and hence this variety was able to quickly grow in the North India. Alright, yes. What is jowar bajra? So you have not had bajra, I don't know what you call in your mother tongue. Just check out what is jowar and bajra in your mother tongue. Alright, yes. Yes, you're right. Great. It is hard to eat North Indian sugarcane, so which means you have tried eating the North Indian sugarcane. Is that so? So that is great, which means you do not need a strong teeth to eat your North Indian sugarcane, but in case of your South Indian sugarcane, you need a very strong teeth because it has a thick stem, right? Millets are not fodders. Actually, millets can be fodders as well. So millets are first edible for humans and they are even edible for animals. All right, yes. So saying south, okay, so remember SO, so south is SO, so Illuminati is giving us an idea. So remember Sacrum Barberry belongs to South India. So great, so thank you so much, right? So we'll stop right here, but we still have to talk about disease resistant and pest resistant. That will continue in the next chapter. So please do remember, so let's quickly go forward so that I will show you the last 2 slides. I thought I'll finish this, but it's okay. So this chapter is to be continued. So we'll have one more lecture. We'll complete this in Friday's lecture. All right. So before we say goodbye, so once again repeating all the announcements. So first announcement is you have a mentee at 6:30. So please do join. At 9 p.m. you have a premiere. So do watch, and it is about your stem cell, and your mentee is about applications of biotechnology. And last but not the least, so tomorrow's class is at 3 p.m. not at 4 p.m. So please do keep an alarm or a reminder. So don't come at 4 o'clock and say, ma'am, hi, and I will be saying bye. So the class has been preponed to 3 p.m. All right, okay, fine. So great. So now it's time to say goodbye. So if you genuinely understood what I was talking about so long, so please do like, share and subscribe. Even if you learned one concept thoroughly, I'm really happy. So please do write in your comments and give me comments, suggestions about what kind of lectures you want in your premiere. I'm always ready for a lot of lectures and classes. All right, so I will meet you in mentee. So prepare well for your mentee and bring in a lot of participants. Yes, thank you so much. Yes, so thank you so much once again, and thank you so much for being a wonderful audience, and this is me signing off, so take care and bye-bye.